Bismillahirrahmanirrahim. Assalamu alaikum. So I welcome you all in my YouTube channel exploring the concepts, and I'm once again with HSSC-I Biology, means FSc first year or high secondary high school. So the topic is today once again the extension of the last lesson, that is light dependent phase, light reaction. And in my today's lesson, I'm going to differentiate between the two different kinds of photophosphorylation, that is non-cyclic and cyclic. Topic is from chapter number 4, Bioenergetics. So let's start our today's lesson. But before that, um, we should go through the objectives of our today's lesson. So my today's lesson is about the terms non-cyclic and cyclic photophosphorylation, also about the understanding of the production of energized compounds through plants. So plants are very, very important as they are responsible for the production of very high energized compounds, which are responsible for the survival of life on land and on earth, you can say. So to understand the difference between cyclic and non-cyclic photophosphorylation is once again another very important question. You need to prepare this in a separate way. So before starting the lesson, we have to go through a quick review from the last lesson. This is basically to revise the last lesson as well. So name the major proteins which are present in the first ETC. So you need to tell me the name of all the proteins which are present in the first ETC, electron transport chain, and that was plastocyanin, cytochrome complex, and plastocyanin. So there are three proteins. And then is what is the importance of the cytochrome complex. So cytochrome complex is very, very important, although all of the proteins which are present in ETC are responsible for the transport of the electrons. That means they are electron carrier, right? But as far as cytochrome complex is concerned, it is not only the electron carrier, it is also acting as a proton pump. So this is very important to note about the cytochrome complex, that it has some specific importance regarding the photosynthesis. So then is what is ATP synthase? It's a knob-like structure present in the thylakoid membrane, and that is basically responsible for the production of ATP during chemiosmosis and involving the membrane, which is thylakoid membrane. So ATP synthase it's an enzyme. How would you define chemiosmosis? So chemiosmosis is actually a combination of three things. That is the transportation of the electrons, movement of the proton pumps, and also the production of ATP involving a membrane called thylakoid membrane. So that what is called as chemiosmosis. So I'm hoping that now you have a good idea about the chemiosmosis and you have no confusions regarding chemiosmosis. It's a chemical reaction, and it is same like that of osmosis. What is osmosis? Osmosis is actually the movement of molecule from higher concentration to lower concentration, but involving a membrane, a semi-permeable membrane. Same is the case with the chemiosmosis and photosynthesis. Chemiosmosis is actually the movement of hydrogen ions from higher concentration to the lower concentration, and this movement also involves the production of ATP. So that what is called as chemiosmosis, right? So next question is, draw the diagram showing the chemiosmosis and photosynthesis. So this is, you have to draw the diagram for the process. So let's start with the non-cyclic photophosphorylation stages first of all. So it has 5 steps. I already have told you, and we have completed so far absorption of light by PS2 and excitation of its electrons. We have completed this. Then photolysis of water, we have done with this as well. And then electron flow from PS2 to PS1. And this is the reaction which is also responsible for the chemiosmosis, right? So next is the two steps we are going to discuss in our today's lesson. And these are absorption of light by PS1 and excitation of its electrons. So now, today we are going to discuss the second photosystem, how it is going to excite and how its electrons are behaving after excitation. And then the last step is electron flow from PS1 to NADP. So the next step is the second electron transport chain, how it works. Right. So let's just start with today's lesson, absorption of light by PS1 and excitation of its electrons. So this is the same diagram I'm using since the whole lesson. So this is PS2, the fourth photosystem. This is ETC, the first ETC, plastocyanin, cytochrome, and plastocyanin. And that is what is called as PS1, photosystem 1, and the second photosystem. And this is going to enter into the second ETC involving another protein called ferredoxin. So this is photosystem 1, the second photosystem, right? Light, once again, in the form of photons. Photons, you all know that photons are just the energy packets. They do not have any mass or charge on them, and they just strike the electron. They just strike the the photosystem one, the cytochrome, not the cytochrome, by the way, the antenna complexes, right? Antenna complexes containing the accessory pigments. One of them is chlorophyll B, and the other is carotenoids. Both of them, they have no direct role in the process of photosynthesis, and both of them are acting as a accessory pigment. And that accessory pigment is actually, it means accessory pigments, they are transferring their energy that they have absorbed um, from the sunlight, and then transferred it to the reaction center. So same is the case. Fluorosystem 1 has the reaction center, and it has also the antenna complexes, right? So photon, through resonance, the energy of the photon is transferring from one antenna complex to the next, and then to the reaction center. And this reaction center has a very specific type of chlorophyll A, which is going to absorb P700 nanometer, just P700 nanometer. Is going to absorb this radiation, and this radiation will, excite the electrons of the chlorophyll, and these excited electrons are taken up by the primary electron acceptor, which is a protein embedded in the reaction center that receives these two electrons of the PS1, P700. And then after taking two electrons, because two photons have strike the chlorophyll, so two electrons are taking from the primary electron acceptor, is going to take two electrons from the chlorophyll A of reaction center of PS1. Means primary electron acceptor is responsible for oxidizing the chlorophyll molecule, and it itself gets reduced. So there is a vacancy creating in this molecule. You all know this is yeah, in this chlorophyll molecule, in this place, by the way, and two holes are creating by the prime electron acceptor. Now who is going to fill these electrons spaces? So obviously there is no photolysis for the PS1, but the two weak electrons that are coming from that yellow line is indicating you the movement of the electrons. So the electrons coming from the PS1 are taken by the primary electron acceptor, then to the ETC, PQ, cytochrome complex, PC, and then finally it enter into the P700 chlorophyll. When it will enter, obviously, when there is a vacancy in the, there is a hole, there is a space, empty space in the chlorophyll A, the space for the two electrons, because everything in this world wants to get some stable position, So same is the case with the chlorophyll. The electrons from PS2 are coming to stabilize the chlorophyll of PS1. Okay, that is how the absorption of light by PS1, and how the excited electrons will move out of the PS1, right? Then is the it will enter into the next transport chain, electron transport chain. Second electron transport chain, I already have told you that the non-cyclic photophosphorylation, it involves two photosystems. This is PS2 and this is PS1. So it involves two photosystems and two ETCs. This is the first ETC, and that is the second ETC. So absorption of light by, so let's go through the text of the topic. 700 in the reaction center of PS1 absorb two photons. Electrons are boosted to a higher energy level. Then 700 molecule passes these electrons to primary electron acceptor of PS1, creating electron holes. The E holes of P700 are filled by the pair of electrons received from P680 PS2 via ETC. So we have completed, we have done the absorption and the excitation and the fulfillment of the PS1 as well. So next is the electron flow from PS1 to NADP. So let's go back to the same diagram. This is primary electron acceptor that has taken two electrons from the PS1, right? Now it has two more electrons, and it is it is disturbed with the two extra electrons, so it at once it gives to the ferredoxin. Ferredoxin itself, it is another iron containing protein. All of these proteins, they have metallic ions, or because they have to work for the transference of the electrons from one to the next within the etc. So two electrons from the primary electron acceptor are entered into the ferredoxin. It means primary electron acceptor gets oxidized and ferredoxin gets reduced. Please these keep these things in your mind, because there may be a question regarding this, right? So then ferredoxin is going to be oxidized by an enzyme called NADP reductase. NADP reductase, it is going to take the electrons from ferredoxin, and NADP positive is present in the system, in the cell. So NADP and electrons, they both of the things, they collected by the NADP reductase and two hydrogen ions. So from where we get these two hydrogen ions? We get these two hydrogen ions from the photolysis. These two hydrogen ions are the same hydrogens they are going to add into NADP. So the NADP, the 2H and the two electrons on their combination in the presence of an enzyme called NADP reductase, because it is going to add hydrogen and it is going to add an electron as well. That's why this enzyme is named as NADP reductase. And the final product is NADPH. So it has an extra hydrogen, as there are two hydrogen. NADP has one place, the stable place for hydrogen. The other hydrogen will be staying with the NADPH. So that is called as non-cyclic photophosphorylation. So let's have a text. The PEA of PS1 passes the photo excited electrons to a second ETC, and the electrons are accepted by FD, ferredoxin, and iron containing protein. NADP reductase transfers the electrons from FD to NADP, and then NADP combines with electrons and hydrogen ions to form NADPH in the presence of an enzyme called NADPH reductase. So we have done with this diagram now. So electron flow from PS1 to NADP. The NADPH will provide the reducing power for Kelvin cycle. Now, this is the point how the electrons are NADP. PS1 to NADP. Electron flow from PS1 to NADP is actually, and plants are responsible for the production of highly energetic and energized compound. And in this NADPH is actually have the reducing power for Kelvin cycle. This is how it works. And the path of electrons through the 2ps during non-cyclic photophosphorylation is called Z scheme. Now, cyclic photophosphorylation. Next step is cyclic photophosphorylation. So cyclic photophosphorylation, if you can recall that, this is PS1, photosystem 1. The cyclic photophosphorylation involves only one photosystem, and it involves only one ETC, right? So it has some differences from the non-cyclic. It involves only photosystem 1, one photosystem, and one ETC. So the electrons coming from the cytochrome complex PC, and it enters into the weak electrons. They have the be the part of the chlorophyll A of PS1, and the two highly energized electrons coming out from the PS1, taking up by the electron acceptor and giving it to the ferredoxin. So they on their routine giving these two electrons to the ferredoxin, but ferredoxin, instead of giving these electrons to the NADP reductase, they gave the electrons to the cytochrome complexes. So cytochrome complex, then the PC. So obviously this energy will be utilized by the plant for the production of ATP, right? And in that case, there will be no NADPH. Why plant is going to do this? Why plant has to do this? Reason behind is the plants have low run of ATP. They are needing more energy, and there is less number of ATPs in the plant. So plant, instead of producing NADPH, which is responsible for the Kelvin cycle, the next cycle, so it is they are, instead of producing NADPH, so they will go for the production of ATP, as plant is needing. Plant has got some infection, or it is under some stress, you can say. Or one thing more, if there is a high increase of NADPH, then in this case as well, plant will go for cyclic photophosphorylation, and they stop for the further production of NADPH. So on the other hand, in the cyclic electron flow, the excited electrons, after leaving a particular PS, finally come back to their PS again for the system. So this is cyclic photophosphorylation on the whole. So this is two, यहाँ से prime electron acceptor, then prime plastocyanin, then cytochrome, then PC, and this. Photosystem 1 get excited through her photon, and then this is the colored area. The colored area is indicating you, by the way, the cyclic photophosphorylation. So let's have a recap of the whole lesson. This is cyclic photophosphorylation. This is primary electron acceptor. Light has entered through the electron carriers, I mean through the antenna complexes. Then it will reaching up to the chlorophyll A, P680, PS2, and then this photolysis will take place, and on photolysis, two electrons will move to the uh, chlorophyll A of our system 2, and oxygen will move to the atmosphere. But we are not using hydrogen at this time. Then photolysis, and then the first electronics first first etc, after moving through the first etc, ATP will produced, and the weak electron will move to the ps1 once again get excited through the like photons, or on all of the antenna complexes. They will move this energy to the reaction center to highly energized electrons get moved to the primary acceptor, and the chlorophyll A of PS1, it oxidized, right? It get oxidized, and it will reduced by who is going to, which molecule is responsible to make the chlorophyll A of PS1 reduced. So plastocyanin is responsible for making the chlorophyll A reduced in its reduced form. So after that, what will happen? The two electrons coming from the PS1 will move to the ferredoxin, and ferredoxin will give it to the NAD to the NADPH. So this is ferredoxin. These two electrons will move to the NADPH. NADPH once again, NADP reductase, NADP hydrogens, which is actually the part of the water molecule, and two electrons coming from the PS1. They all on the combination of all these three things, NADPH will form, which is a assimilating power, or a very high energetic compound is produced through the second electron transport chain, and that is called as NADPH. So another very important thing regarding this top chapter is the difference between non-cyclic and cyclic, right? So non-cyclic has using both the PS1 and PS2, right? But in cyclic, only one photo system is used, right? Photolysis will take place in this reaction. No photolysis will take place. Water is consumed. Water is not consumed. Oxygen is liberated. No oxygen is liberated. Source of electron is water. Source of electron is P680. So it's 700. They have written there 700, but obviously not. This is P680 coming from the electrons coming from the P680, which was PS2. And then it is affected. There is no need to write this reaction. This is actually their confirmatory test, and it is found in green plants. It is found in bacteria and green plants. And NADPH is also formed along with the ATP, but in this case, only ATP is formed. And one more difference is this cyclic will plant will go on for the cyclic when it is under the stress, when it is under some infection, it is some under environmental stress. So plant will go for the production of ATP. As there is low run of ATP, it has to survive first, then for the production of other molecules. So that is all from my side. The difference between the cyclic and non-cyclic, a very, very important question. This is itself a very important question. Chemiosmosis is very important. So if you have any question, you can write me in the comment box. We will meet again in the next lesson. Up till then, Allah Hafiz.